Good afternoon, George, and welcome to this presentation, also the rest of the class. Today we'll be discussing visual sensory physiology with specific reference to vision loss and the development of visual protheses, also known as the bionic eye. I'll be mainly focusing on retinal-based implants, and the first of which was successfully trialled in humans in 2012. Now, in order to understand the advances in technology around this, we first have to look at the normal physiology of the eye. So let's go. The eye is one of the most complex and specialized sensory organs ever to have evolved. Given this, I could literally spend the next 6 minutes just discussing its normal physiology. However, in general, it consists of a number of structures that focus light onto the back of the eye, called the retina. Now, the retina lines the posterior 3/4 of the eyeball and consists of two layers. One is the pigmented layer, second, the neural layer. Now, let's take a quick look at the visual pathway. Excited by light energy, the photoreceptors are in synaptic relationship with bipolar cells. These, in turn, interact with the third neuron in the visual pathway, called ganglion cells. Now, ganglion cell neurons converge to form the optic disc and in turn the optic nerve. In this way, light energy is transduced into electrical impulses which course the optic nerve to the thalamus and then go to the primary visual cortex, which is the vision center of the brain. It is here that the signals are interpreted and we perceive vision. As of 2016, more than 450,000 Australians are impacted by vision loss or blindness. Two causes of vision loss include age-related macular degeneration. An example of what a sufferer would see is shown in the middle image. The second is retinitis pigmentosa, and an example of what a sufferer of that would see is shown in the bottom image. Age-related macular degeneration is a name given to a group of retinal degenerative diseases which cause progressive loss of central vision. Now, this disease impacts the retinal pigment epithelium, which acts as a barrier and passes oxygen and nutrients to the retina, as well as taking waste to the highly vascular choroid. Now, in macular degeneration, the RPE cells start to accumulate a whole host of waste products, and this aggregates over time to form yellow sacs under the retina called drusen. Loss of vision in age-related macular degeneration occurs due to photoreceptor cell death as the retinal pigment epithelium degrades over time. Retinitis pigmentosa, on the other hand, is an inherited disease which causes harmful changes to any one of 50 genes. These genes code for a number of proteins which are essential to the healthy functioning of photoreceptor cells. Now, this disease results in the death of photoreceptor cells, but most of the other retinal neurons remain intact. Retinal-based bionic eyes are a visual prosthesis technology which is suitable in both age-related macular degeneration as well as retinitis pigmentosa. This is because both of these conditions result in vision loss due to the death and destruction of photoreceptor cells, whereas the optic nerve and many of the other retinal cells remain largely intact and functional. Sufferers of these two diseases, although they have vision loss, essentially most of the hardware in the eye is still there and functioning. The bionic version of the visual system consists of glasses on which a camera is mounted, as well as a body-worn processing unit, which is no larger than the size of a mobile phone, and the electronics to send these signals into the visual processing pathway in the human body. So how does this system work? Well, the camera picks up visual stimuli from the environment and transmits this data to the processing unit worn on the body. Here, the information is processed and then sent via an external wire to the receiver device implanted in the patient. From here, the signal is sent to the retinal implant, where a series of electrode arrays converts this signal into electrical impulses, which are then picked up by the remaining intact retinal cells. From here, the message is sent normally down the optic nerve to the visual processing center of the brain. This electrical stimulation of the remaining cells in the retina caused the patient to see small spots of light called phosphins. This is a phenomenon of perceiving light without light actually entering the eyeball, similar to when you close your eyes and you can see the different colors. Now, vision gained through the bionic eye is not like normal vision. It consists of a series of spots that must be interpreted through training in order to visually map out the environment for the patient. Through the use of retinal-based bionic eye technology, sufferers of both macular degeneration and retinitis pigmentosa are able to regain a sense of vision, allowing them to navigate their surroundings and perceive their environments. Thank you so much for listening to this presentation, and I hope you've learned a lot.